All right, welcome everyone. I just want to introduce the presenter of the next workshop. Chloe Mirzayi is a PhD student of epidemiology in my lab. So she has a really interesting set of interests and expertise spanning bioinformatics and epidemiology. And she has a special interest in causal inference, which is an area that many bioinformaticians perform analyses in, even if they don't know that they are. So anyone who has done a multivariate model to control for confounders has done causal inference, even if you haven't had any training in it. So the idea behind this workshop is to give all of you a better idea of the underpinnings and execution of these kinds of analyses. So with that, I will hand it over to you, Chloe.
Thank you so much, Levi. Hi, everyone. My name is Chloe, as Levi mentioned, and I work with Levi. So for today's workshop, I'm going to be talking about epidemiology for bioinformaticians. And, when I gave this talk a year ago, I felt like I had to introduce epidemiology to the world, and maybe people hadn't heard as much about it. But I think with COVID-19, epidemiologists have been very present. You've heard a lot about epidemiology in the context of the pandemic. And I want to acknowledge that a key part of epidemiology is disease surveillance and pandemic modeling, which is probably what you're more familiar with right now. But modern epidemiology also has a focus on elements of causal inference, including, how do we decide that something causes a disease, as well as related concepts such as research design and how we can effectively answer research questions related to human health using research design, using causal inference. And so, yeah, so you might have heard in the news about epidemiology, but we'll be focusing more on causal inference. So today's workshop will start by, I'll talk about what is causal inference, what is a cause, what do we understand about that. Then I'll look at some common sources of bias and confounding in research studies. Then we'll talk about study designs, so some common study designs and how causal inference operates within those types of study designs, and how do we develop a causal argument within those types of studies. I'll introduce some packages available in R to help with causal inference and for diagramming out the assumptions and the ideas you have. I'll show a worked example in curated metagenomic data of kind of starting with a research question and then moving on into considering my assumptions and then looking at the results. And then finally, I'll have a few problems that we can talk through or work through, and then we'll wrap things up. So with that, I'll get started. So, and I should mention also that if you have a question or if you have something you want to discuss, just you can place it in the chat or polls section on the Pathable website. And Nitesh is monitoring that, and he can relay any questions or comments you have. So what does it mean to cause something? It's a philosophical question. What is a cause? And I thought about this a lot. It's not easy to put it into words. I would say, an event that precedes another event that brings about that event, or something that results in something else occurring. But that's not very scientific. It's not very descriptive, and there's not something testable there. But despite that, I think humans, animals, we all have a very good intuitive understanding of causation. I know that when I turn on a light switch or press a key on my keyboard, that I'm causing something to happen, and usually, barring any, third variable, I know what the result of that causal event is going to be. And so, when I burn my finger cooking, I can trace back the causal pathway kind of intuitively and say, okay, what caused my finger to come in contact with that hot stove? perhaps it was me being careless or me being distracted is a primary cause. And then I could go even further backwards and say, okay, but what caused the stove to be on in the first place? Well, it was me turning the stove on. And why did I turn the stove on? Well, even further backwards, it was my motivation to cook dinner. And so even though causal inference sounds very big, I think we actually have this intuitive understanding of it. And in fact, as in this example I give here, my cat, he's learned that, if he jumps on the kitchen table while I'm eating, the dinner I just prepared after burning my finger, I will spritz him with water bottle because he's not supposed to be up there. So my cat could be considering kind of the causal variables here when he decides to jump on the table. He could be considering, is the spray bottle nearby? That's a variable there. Am I eating dinner? Am I distracted by something? So there's actual, like animals and humans really intuitively think about causality, but that doesn't help us as much from a scientific perspective. Like we want to be able to ask a scientific question that we can test with a hypothesis. And so that is where the counterfactual definition of a cause comes into play. A lot of our work in bioinformatics and in epidemiology is asking questions where we can't observe it directly. We can't, there isn't always a hot stove or a cat jumping on a table. Instead, it's happening on a genetic or a microbiome level that we have to, collect lab samples, and we have to collect a whole study to try and observe a causal event of interest. And so we must draw inferences based on data. And so what we have come up with is what's called the counterfactual definition of a cause, which allows us to make a testable hypothesis, which is what would have happened had the event of interest or what would have happened had the event of interest not occurred. So if we've got some causal variable, if that causal variable had not happened or happened differently, what would we have observed instead? I also like to think about this from like the science fiction sort of thing. So imagine an alternate reality where everything is the same, but this one event of interest happened differently. And if we can contrast that and say, okay, well, if this person had lived the exact same life, but hadn't smoked, would she have still gotten lung cancer? And so, then we contrast. So if they smoked and they didn't smoke, that's the only difference across all these across this different reality. And then we could say, okay, well, the only thing that changed was their smoking, and therefore we're pretty confident that's what caused lung cancer for them. But let's consider another kind of more fun example. So imagine a dog at home and there's an ambulance that drives by with its sirens on. And the dog starts barking. And we can ask a causal question in a very straightforward way. And in this sense, it's a research question. Did the ambulance driving by the house cause the dog to bark? And we could come up with a hypothesis from that. We could say, yes, the ambulance driving by did cause the dog to bark, and then we can try and test that. But we can also rephrase that question in terms of the counterfactual. Would the dog have barked if the ambulance had not driven by the house? And then we can visualize this relationship. So make a diagram of this relationship, sometimes called a causal diagram, also called a directed acyclic graph, or a DAG, a DAG, a DAG. And so we've got our DAG here for this relationship that we're interested in. So we've got the ambulance driving by, which we hypothesize causes the dog to bark. And now, unlike maybe a statistical relationship or a non-causal relationship that we're interested in, maybe an association, when we create a diagram of a causal relationship, we want to denote what's the cause and what's the outcome. So in this case, we've got an arrow pointing from ambulance to dog barking because we believe the ambulance caused the dog to start barking. So in all DAGs, causal diagrams, we must state a directional relationship between each variable if we believe a relationship exists, because we are theorizing that one variable causes another. So in this case, the ambulance is my cause or my exposure or my treatment, and then the dog barking is my effect or outcome or condition of interest. So this diagram that I've done here, which is two variables, is very simple. Obviously in the real world, there's a lot more probably going on than just an ambulance and a dog. Um, and if we were to if we just wanted to establish, to try and measure the causal effect of the ambulance on the dog barking, what we would do perhaps is do a study where we, randomly drive by some houses with dogs with ambulances, have some of them, or have some houses have ambulances drive by with the sirens on, some houses not, and then see if the dogs bark and see if there's a difference. And so this would be a very simple, we create a very basic regression model. Our independent variable would be, like 1, 0, a siren from an ambulance, and then our outcome would be 1, 0, a dog barking. However, we're not really that lucky in health research that there's just two variables. And there could be a lot of sources of bias and confounding to this relationship. And so my question is, can you think of any situations that may bias the observed relationship between the ambulance and the dog barking? And just off the top of my head, I would think, yes, there could be, maybe there's something else that caused the ambulance to be nearby and the dog to bark. It could be that we only noticed the dog barking when the ambulance arrived because that woke us up, but perhaps the dog was barking before the ambulance arrived. So there's a lot of different possibilities that could possibly cause us or to observe this relationship when in fact no relationship exists. And so these variables are, or this could be bias and confounding. So to start with, I'm going to talk about confounding, which is very common. Most people who work in bioinformatics I think have an understanding of confounding, but I will explain it from an epidemiological point of view. And so confounding arises when there is a third variable in this relationship between cause and effect. And specifically, there's 3 criteria that have to be satisfied for a variable to count as a confounder of a relationship between an exposure and an outcome. The confounder must cause the outcome. The confounder must cause the exposure, and the confounder is not a mediator. That is, it's not kind of present on the causal pathway between the exposure and the outcome. Therefore, it's a third variable. It's not mediating this relationship. And I really want to stress that it must be causal for the outcome and the exposure. So it's not sufficient that it's associated with them, but you have to believe that it causes them. And we'll see why that's important in just a moment when we talk about colliders. But if we go back to our example, as I mentioned, there could be some third variable that causes the dog to bark and causes the ambulance to be in the area with its sirens on. And so in this example, we can think of a car crash, which is the ambulance is responding to the car crash. The sound of the car crash alerted the dog and caused the dog to start barking. So if we just were looking at the 2 variable model and we weren't aware that there had been a car crash, we might falsely assume that the ambulance caused the dog to start barking when in fact the car crash was really the main driver of that. Or perhaps the ambulance, did cause the dog to bark, but the car crash also magnified the amount of barking that the dog did as a result of this. So to go into kind of a more health-focused example, again, when we're thinking of smoking as our exposure and lung cancer as our outcome, in the 1950s, a lot of skeptics said, okay, but what if there's some genetic factor, some, genetic variant that both causes people to smoke and causes lung cancer? And so they argued that there must be a lurking confounder that was a genetic component um, that was causing both. And so we do in fact know now that there are I think a few genetic variants that do cause both smoking and increase the risk of lung cancer. But what the researchers at the time did was that they looked at what the magnitude of confounding must have been to to create the observed relationship that they saw in smoking and lung cancer, and found that that genetic variant would have had to cause it was something like on the order of a 900% increase in the risk of lung cancer, which and they were able to disprove that genetic variant alone could not account for the relationship they were seeing between smoking and lung cancer. And so this pathway between the exposure and the outcome that runs through the confounding variable is called a backdoor pathway, essentially because we've got this front door pathway which runs from the ambulance to the dog barking, but then we've got this backdoor path that runs from the ambulance through the car crash, through the dog barking, and that biases the relationship between the exposure and the outcome we observe. And so what do we do when we have this backdoor pathway? Well, we want to close it off. We want to stop this pathway, from biasing our result. And the classic way of doing this is to stratify your data by levels of the confounder. So what we would do in this scenario is that we would look at ambulances that responded to car crashes and then dogs barking, and then we would look at ones where perhaps the ambulance wasn't responding to a car crash, it was just driving past on the way to the hospital. And so we could then stratify and then look at the effect in each of those strata and see if we still see an effect there. And that would give us evidence that we would control for the effect of the car crash while still observing the effect of the ambulance on the dog barking. And in a more modern regression model, we can include the confounder as an independent variable in the model alongside our exposure. And when we include the confounder as a variable in a regression model, we are controlling for it or deconfounding for it. Obviously, how you model that variable in terms of, if you model like as a linear variable or categorical, that is still a statistical question, whereas, so you do have to model the variable correctly, but for causal inference purposes, as long as we have modeled the variable correctly and it is entered into our regression model as an independent variable, we have effectively blocked that pathway. Now, in contrast to confounders, there are colliders. So taken at, a glance, colliders appear to be very similar to confounders. But whereas a confounder is a common cause of the exposure and the outcome, a collider is a common effect of the exposure and the outcome. So that means the outcome causes the collider, the exposure causes the collider, and again, the collider isn't a mediator, as was the case with confounders. And so again, in our example, we can see ambulance and dog barking both result in sleep disruption for nearby neighbors or residents of the home. And so in this case, sleep disruption is not causing the exposure of the outcome, it's being caused by the exposure and the outcome. And again, we can see the difference here where we've got our car crash example where we can see that the arrows point in different directions. And adjusting for a collider as one would a confounder can actually create bias. In effect, by adjusting for collider, that backdoor pathway, which we talked about in the confounding example, gets opened in the collider example. And so why is there no backdoor pathway in the collider example? Because when there are two arrowheads pointing at each other in a DAG or in a causal model like this, that pathway is blocked. So the pathway here between ambulance to sleep disruption and dog barking is blocked because these two arrows intersect here. And so that backdoor pathway is blocked unless we adjust for it. So if we were naive and entered sleep disruption into our model, because we thought it was a confounder of this relationship, we would effectively create bias. We would create a backdoor pathway between ambulance and dog barking through sleep disruption. And so the correct way for dealing with colliders is to ignore them. Hypothesized colliders should not be adjusted for or included in models. But this becomes an issue because a very common form of, this is what I was saying about arrows being blocked, but a very common form of collider bias is selection bias. And so selection bias is a type of collider bias, and this emerges when selection into the study is dependent on the exposure and the outcome in the study. So again, this is a collider, and the issue here is that the backdoor pathway through selection is opened as part of the process of selection into the study. And so to give an example of where this becomes an issue, one of the professors that I work with calls selection bias the already dead problem, which I think is very memorable because people who have already died of the outcome are not alive to be in the study. And these people could have a very serious or aggressive form of that disease, and not including them biases the causal relationship. And so you'll see selection bias referred to by a lot of different names, sometimes in particularly in workplace related research. It's called the healthy worker bias, where people who are unhealthy, drop out, leave that workplace because they're not doing well. And this often comes up in studies of people who work, in like mines and factories, that they'll often report that, well, our workers are very healthy, and that's because all the unhealthy workers have already left that job because they're unhealthy. But you also see it in randomized control trials because loss to follow-up is a form of selection bias. If it happens and it's dependent on the exposure and the outcome. So we could think of perhaps a drug that is given to people for a certain disease, and then we measure their symptoms over time, and perhaps the side effects from that drug worsen the condition for some people, and those people drop out of the study, and we don't get data for them. And so we don't know, why they dropped out of the study, and we have in fact selected for people who only responded well to the treatment, and people who didn't respond well to it didn't complete the study. And so loss to follow-up is a very important source of bias, and unfortunately, it becomes really difficult to account for this bias because part of the issue is that these people have dropped out of our study, and we don't have data on them necessarily for the outcome, or we have incomplete data, or they weren't around to even be in the study in the first place, so we have no data on them. And so accounting for this type of bias is very difficult. And so a very common question is, what do we do about selection bias? And the answer is, unfortunately, try and design your research study so that you can minimize selection bias. There have been some attempts by studies, especially RCTs, they try and follow up with people who refuse to complete the study to see if they could collect data on why they refused to complete the study. For instance, why did they stop taking the drug? Why did they not come to any follow-ups? Why did they refuse additional lab work? And then one other thing you can do is sensitivity analyses. So you can you can say, okay, well, if I lost, let's say, 10% of my sample due to, them having severe side effects or due to them not wanting to continue to participate, how would that change my results? And so those are some of the main methods for addressing selection bias. Unfortunately, unlike confounding, where we can just include it as a variable in the model, if we have those data, selection bias is a bit more difficult. Um, Okay, so in this next section, I'm going to talk about different study designs and then what they are indicated for causal inference, and then what sources of bias or confounding that they can have in these types of studies. So randomized control trials, as probably most people know, are the gold standard of research because the exposure of interest is assigned at random using, a coin flip or a more advanced technique. And as a result, because we've assigned that exposure at random, there are no possible confounders of the relationship between exposure and outcome. Remember that one of the criteria for a confounder is that it must cause the exposure. But if the exposure is assigned at random, there can be no other variables, no more arrows pointing at the exposure. There can be no other causal variables for that exposure, assuming the randomization was done correctly and was not biased. And for that reason, RCTs, as we know, are the gold standard for causal inference. And often we are confident taking the results of an RCT and interpreting that as a causal finding. However, selection bias is still possible in RCTs. As I mentioned, loss to follow-up is possible in an RCT, and when we lose people to follow-up, that could bias the observed causal effect of the exposure on the outcome. And so even though this is the best type of study design we have for causal inference, it's still not immune to bias. And that's something we need to keep in mind when we're interpreting an RCT. And often when I'm reading an RCT, I ask myself, a manuscript of an RCT, I ask myself, okay, where could selection bias occur in the study? Did they report how many people they lost to follow-up? Did they report any attempts to retain those people or any sensitivity analysis based on that loss to follow-up? So a related design to an RCT is an instrumental variable IV analysis or Mendelian randomization, MR. And this attempts to mimic an RCT using a source of kind of pseudo randomization. So if we look at the DAG for an RCT, we've got a coin flip or a randomizer that assigns an exposure, and then we follow up with them over time to see if they develop an outcome of interest. And so nothing, the C here is a little misleading. There's nothing that can possibly cause this this exposure since we assigned the exposure at random. And so effectively, we've blocked all potential confounders of the exposure and the outcome. In IV or MR, in instrumental variable or Mendelian randomization, the coin flip is replaced with a source of randomization, such as genetic variation. And so in this case, we look at, a genetic variant that we assume is more or less randomly distributed, and we can use that as a causal variable for our exposure, which then we follow up over time to see if they have the outcome. And so Mendelian randomization is common and hopefully better than just an observational study in establishing a causal effect, an estimate of that causal effect. However, it is you have to assume that your kind of pseudo-random element here is truly random. and there's still the possibility of selection bias. And then I just wanted to point out there's also other types of instrumental variable studies where they look at before and after implementation of a new government policy as kind of the randomizer. Another one that's done is distance from a medical center. There's been some studies that show that like the further you have to travel to a medical center can be used as kind of a pseudo random variable, which is interesting. So next I'm going to talk about observational studies. So observational studies, as we know, opposed to RCTs, we don't assign the exposure. So we just follow up with people over time. So cohort studies are a type of observational study where the participants are chosen on a common characteristic, such as occupation, location, or history of a disease. There are some very famous cohort studies. Probably the most famous is the Framingham Heart Study, which is the common characteristic is that they're all residents of Framingham, Massachusetts. And so all the participants or all the people in this town are followed up with over time for various heart outcomes to see, if they develop heart disease. Um, and then you can measure exposures and outcomes over time, and then see, you can contrast them. And hopefully, the thought is that these people are pretty similar. They all live in this town, and the idea is that hopefully, people who are effectively neighbors, maybe neighbor one develops heart disease, whereas neighbor two, who's the same age, similar socioeconomic background, doesn't, and that allows us to create that kind of contrast, that counterfactual, where we've got two very similar people, and the only difference is one developed an outcome and one didn't. And so we can look backwards in time and then see what was different about them, and hopefully start establishing kind of a causal argument. However, because this is an observational study, both selection bias and confounding can occur. So, we did not assign the exposure at random, and so it can be confounded by any number of variables. And in fact, they try and measure as many confounders as they can, but there's always the risk of an unmeasured confounder because they can't measure every single variable of a person's life. Another type of observational study is a case control study. The difference here is, whereas a cohort study, we start with a group of people and just follow up with them over time to see their exposures and outcomes. In a case control study, we start with people with an outcome of interest, the cases, and then we try and match or find similar people to those cases who are as similar as possible, except that they don't have that disease or condition of interest. And so I see this quite frequently in microbiome research, where they will take, let's say, like 20, 30 people with diabetes, and then they'll find 20 or 30 people without diabetes who are similar in age, sex, and other socioeconomic variables, and they'll try and match them. And the idea is that these people are very similar, but for having that disease or condition of interest. But as with cohort studies, selection bias and confounding can occur. We can't match on everything. And even if we match on a lot of variables, there still can be other very important variables that are harder to measure or assess in terms of that person's life. And so again, there are sources of bias in case control studies that may affect the causal relationship we observe. And a final type of observational study is a GWAS. So this is a genome-wide association study. I just want to highlight that this is an observational study, and participants are chosen on a specific phenotype or disease. And a GWAS can be a case control study, or it can be a cohort study. But as a result, we know that it can be subject to selection bias and confounding can occur in a GWAS just as it can in other observational studies, which is something to keep in mind when we're interpreting the results of a GWAS. And And so the question is, what are some possible sources of confounding or bias in a GWAS? The one that comes immediately to my mind is batch effects, could confound the relationship if especially if certain samples were done in different labs. For instance, I think I saw an example once where cases where people in a hospital setting, and therefore the lab that ran the samples was in the hospital setting, whereas people from the community were the controls, and their samples were run in a different lab, and that created a source of bias because they were being run in two different places. So that's a quick summary of the different study designs, the limitations of them, and how we can interpret them. So I want to point out also that I think it's very easy from a causal inference perspective to leave this and interpret this as well. We're never quite sure. You know, all these study designs are potentially subject to bias and you know, the observational studies are particularly vulnerable to confounding and unmeasured confounding. And so we really can't ever say that anything causes anything. And I've had that feeling at times, but I think what I want people to take away from this is not that, you know, you can't prove something causes something. It's just that when we interpret things, we can make a causal interpretation, but we just need to be aware of the limitations and the assumptions that we're making as we draw this interpretation. And for that reason, that leads us into our next section, which is causal inference in R. So I'm going to just discuss a few of the packages that I use when I'm thinking about causal inference in R. And then how do we go from you know, making our initial hypothesis or causal diagram and then actually interpret that, and that's when we'll do the example. So the first is Daggety. So all these causal diagrams that I've been making, these DAGs, I've been making using Daggety. And it's really straightforward, and I really love it because you just put where the arrows go, and you can create pretty complicated DAGs pretty simply, and then you just plot it. This one's a little more complicated than the ones I've shown you so far, and it can do very complicated DAGs or simpler ones. And I think it's pretty easy to parse. And then the nice thing about Daggety as well is that we can say, okay, I want to look at the effect of X on Y, you know, try and interpret that from a causal perspective. Assuming that this DAG is correct and that I've modeled all the variables that I think have an impact here, what do I need to control for to find the effect of X on Y? And we run this little snippet of code here, which gives us what they call the adjustment sets, or the variables you would need to adjust for. And then we get this nice output here, which tells us for each relationship between two variables, what we would need to control for. And so we can see X on Y down here, where it says the effect of X on Y is identifiable, controlling for W2 and Z2, so those two variables, or we can control for V and W2, or V and W1, or W1 and Z1. So let's just take a look, W2 and Z2. So up here in this model, if we're interested in X and Y, all we would need to do is control for W2 and Z2. And we would effectively have adjusted for potential confounders in this relationship, and we could identify the effect of X on Y. And so even though, you know, your DAGs can get quite complicated, R can just tell you what you need to control for. And so I think this is important, especially because I think if you're doing this by hand, it's sometimes you try and like avoid making your DAG too complicated because you don't want to try and figure out where all the lines are going and what it all means. But if you do this in R, it can just tell you what you need to control for it in the end. So you can make your model as complicated as you want it to be, or as you believe it to be, based on your knowledge of the relationship that you're interested in. Um, built atop of building on Daggety is ggdag, which allows you to make a little bit more beautiful and more customizable DAGs using ggplot2 kind of language. And so in this case, I just asked it for what do I need to adjust for in this same model as up above to find the effect of X on Y. And I get this nice diagram that says, you know, blue variables are variables I don't need to adjust for, red are variables I need to adjust for, and then it gives me 4 different adjustment sets. So the same thing I got above in this text kind of response, I'm getting now in a graphical format. And we can see, you know, W1 and V1 or V are variables we can control for, and we would get the effect of X on Y. And so ggdag is really nice, especially for making figures for publication, as we know, ggplot2. Now, in terms of once we've actually created, you know, kind of our diagram or causal model, what we then want to do is adjust for the variables that we have identified in our adjustment sets as variables to adjust for. And so we can do that in a lot of common bioconductor packages. So in DESeq2, we can just, when we create our DESeq data set, we can specify the confounders as part of our design. Edgejar and limma have a model.matrix function, which you can use to add in a confounder. So for instance, here we had like a batch effect. And then we can model it using that design. And so let's actually do this. So what I'm going to look at is using curated metagenomic data, which is a package that Levi and I work on that has a bunch of microbiome and metagenomic data sets that are all curated and analysis ready. So it's perfect for this. So the first step, I'm going to think about my assumptions and my research question. So I'm interested in looking at the relationship between colorectal cancer and altered microbiome. And specifically, I would say I hypothesize that colorectal cancer causes an altered gut microbiome in humans. And so I would start by saying, okay, well, what else could possibly confound this relationship? And I would say age probably confounds it. Your microbiome probably changes with age, as well as, you know, your risk of colorectal cancer probably increases with age. And with that, gender probably affects both as well. And so I can create my DAG here. And so I can see that I've got colorectal cancer. I don't know why I put it on the right here, but colorectal cancer is my exposure, microbiome as my outcome, and then 2 confounders, age and gender. Obviously, this is very simplified. I think in reality, I would theorize that there were a lot more confounders: diet, exercise, other lifestyle factors, stress, probably pretty important as well. But this is a simplified example. And so then we can just say, okay, what do I need to adjust for? And again, I just asked ggdag to give me the adjustment set. And it tells me I just need to adjust for age and gender. And so now I feel like I know everything that I need to know to actually run my analysis. I know what I need to adjust for. I have my question, my research question. I've got my hypothesis, and now I just need to model it. So I download the data from curated metagenomic data, and then I take a look at it. Again, I would probably do a lot more descriptive statistics in real life. I would, you know, look at the distribution of age, the gender, look at a lot of different variables, create a descriptive statistics table. But for now, we see that we've got 52 controls and 52 colorectal cancer cases. And then first, I'm going to do a model, just a crude model, where I look at the exposure, so colorectal cancer, and the outcome, which is differentially abundant microbiota in in this study. And so I do that using DSeq. As you can see here, my model is just study condition is my only independent variable. I'm using an alpha of 0.10 just because I'm exploring and I just, want to be pretty lenient. So I get 5 significant results. So these 5 bacteria are differentially abundant between cases and controls, not controlling or adjusting for any of my potential confounders. So my next step, so now I've identified these 5, I want to see, okay, well, let's add in one of my confounders and see what changes. So now I'm going to add age. I'm using categorized age, but you could use continuous age or age squared, depending on how you believe the relationship between it and the dependent variable to be. And we run it, and we, run the same thing we did before, but now we just have age category as an independent variable. And we see we only have 2 results now. So 3 of our results are no longer significant once we've adjusted for age. And so that potentially indicates that we only saw those results due to confounding by age. And finally, we add in gender, and we do the same thing. So we include age, and now we've added gender. And then we look at the results, and we see, again, it's the same 2 significant results. So it appears gender didn't change much. So the question that comes often here is, well, gender didn't really seem to change anything. Can I remove it from my model? And the answer to that is probably not, because if you hypothesize something to be a confounder, whether it's statistically significant or not is irrelevant, because we want to test the model that you've created, and whether that was statistically significant or not is not relevant to the research question I'm asking, which is, is this effect significant when adjusting for age and gender? And so, yeah, so we have deconfounded this. We have for age and gender, and we have got our results, and yeah, so that's how I would do a basic kind of starting with drawing my assumptions and then looking for what I want to adjust for, and then creating my model, and then I would then report my results. And so that's a very basic example of how I would kind of bring this causal inference thinking into a real world example using curated metagenomic data. And then I have some toy problems. We don't necessarily need to discuss these now, but there's something that you can try out on your own to think about what we've talked about here today. So in this first one, I give you this very mean-looking DAG and ask you to figure out what do you want to control for if you want to find the effect of X on Y. You can try creating your own DAG and identify what variables you would need to control for to measure the causal effect of interest. And then I have a question about transportability. And so to wrap things up, unfortunately, there's no silver bullet for confounding or selection bias or collider bias in our bioconductor. But instead, we as researchers have to think through our research question and the assumptions in that research question, drawing a diagram, a causal diagram or a DAG of that relationship that we're interested in, and then trying to identify potential sources of confounding and bias using that diagram is very helpful. And as one epidemiologist even says, draw your assumptions before you draw your conclusions. Obviously, unmeasured confounding is really difficult to deal with. It's a difficult problem for causal inference, and we haven't measured the confounder, so how can we adjust for it to deconfound for it? That is a good question. Something we could do is sensitivity analysis. Oh, did you lose me again? My internet connection is unstable, is what
No,
it
you're
says.
fine.
Oh, perfect. Um, and it's just important to remember that the definition of a cause is rooted in that counterfactual, and we need to find a way to using our research to come as close as possible to observing what would have happened if a participant's exposure status has been different. So trying to find that alternate world and figure that out. So that's my presentation. Thank you. I'll open up to questions. I also have some further reading materials here. So if you want something easier to read and something like a fun summer read, I highly recommend Pearl Mackenzie's Book of Why. That's written in kind of a very beginner-friendly format. There's very little math. Hernan and Robbins have published a full causal inference textbook online, which is very thorough, has a bit more math, and you know, kind of explains the theories behind a lot of what I told you today. If you really want to challenge yourself, Pearl wrote a book called Causality. It is incredibly difficult to read. He's a computer scientist, and um, it's challenging, but yeah, it's brilliant but challenging. So if you want a challenging and but very thorough read on causality, that book is excellent. And then Hernan launched an edX course, which is free online, which allows you to learn a lot more about causal diagrams, the information embedded within those diagrams, and how to interpret them and use them in your research. So thank you, everyone.
Thank you, Chloe. We have a whole bunch of really interesting questions that there probably isn't enough time to get to, so maybe I'll choose a few for you, but you might want to go into the, somehow add these as a, you could answer them in the poll window, or even add them to the end of the workshop or something. Some of them just as a Q&A, they're really interesting. Anyways, the most popular question was, is the confounder effect similar to the batch effect? And that was something I had wanted to ask you about too, is whether you agree with the use of confounder to refer to batches, and with the way that they're often included as in a multivariate model. And also, can imputation take care of selection bias?
Both are excellent questions. I think batch effects can be confounders. Obviously, it has to depend on the exact nature of the relationship of how the batches were run. If especially if the batches were dependent on the exposure or the outcome, I think that can create a type of confounding bias. I think, again, it's helpful to draw the DAG and think this through, because I think when I think it through, it's also maybe a type of collider bias, potentially, or selection bias, where if different samples are going to different labs based on the exposure of the outcome, that's maybe more selection bias. So I think it's helpful to draw the DAG and think through where you think the bias occurs and how that bias is your result. There's also, I didn't mention measurement bias, but measurement bias is also can be important. And I think that could be a type of batch effect bias in terms of, they call it like differential measurement bias, where the magnitude and direction of the bias can be dependent on either the exposure or the outcome, hence why it's differential. So that could be that batch effect. But I think controlling for it is, you know, as a kind of confounder or as an independent variable in your model is a correct way to deal with potential batch effects.
Maybe I could just add to that, because I've seen your workshop before and learned this
Oh, yeah.
from you, so I'm not adding, but Daggety has a web page too, where if you're not sure if you should control for something, you draw things out, it gives you R code so you can put it in your paper, and it tells you what you should put in your model, which is amazing. And entirely, I thank Chloe for teaching me that.
Yeah. I could pull that up real quick. So if you
Maybe
just,
while you are, you could answer that other question too of whether imputation can take care of selection bias.
uh, I think there have been some people who have proposed that imputation can be used for selection bias. I think the issue though, again, is that these data, I think one assumption for imputation is that the data are missing at random. Uh, Correct me if I'm wrong on that because it's been a moment since I've done missing data. I think selection bias means the data would be missing not at random or not missing at random, which would then violate one of the assumptions of multiple imputation. Um, because selection bias is due to some kind of, uh, you know, causal effect between the outcome and because it's a collider bias, so the exposure and the outcome are causing the selection bias to occur. Um, Therefore, the data are not missing at random. Yeah, which is one of the assumptions for multiple imputation. So I think there have been some attempts at trying to see if you can impute missing data and see if that will help with selection bias, but I think the most common thing I see for trying to account for selection bias is sensitivity analysis, where they say, okay, well, we lost this percentage of participants. If all of them had the worst possible outcome or the best possible outcome or, a moderate, somewhere in between, how would that change my findings? Um, and I think that's the way I've seen for dealing with selection bias, but there's other methods out there, and there's a lot of new ideas because it's such an issue and a lot of research. And so real quick, this is Daggety. You can just find it by searching Daggety on Google, Daggitty. And yeah, it's really simple. You can just say, I've got my exposure. So it's say smoking. Let's say my outcome is, microbiome. And then I can just click on the variables. And then I've got my exposure, my outcome. And then if I want to add a confounder, I just click to add another. And let's say, let's call it confounder. And then I can add the arrows between my exposure and my outcome. Oh, whoops. So I want to specify this as my exposure. Whoops. And this is my outcome. And now we see this is labeled red because it's a confounder. And then if we, here we've got our model code. You can also, where's my export to R? They moved things around since the last time I was here, but there is a way to, you can export the latex. There is a way to export it to R as well. Yeah, anyway, so Daggety is really useful. You can also just do this directly in R if you prefer.
All right, we have to let people get on to the next workshop, but let's make sure that you have all of the questions that appeared in the poll, and I'll recommend that you maybe add them to the README file or the vignette as a frequently asked questions so that people could check in on them later.
Oh, wow, yeah, I see all the questions. Oh, they didn't load at first.
Lots of questions.
Wow, okay, yeah, I will definitely
Okay.
try and answer them all. Yep, thanks everyone.
All right.